cuts the regulatory interspaced short palindromic repeats, or CRISPR, with the associated proteins called Cas proteins. Authority discovered as an adaptive immunity mechanism in bacteria and archaea to counter bacteriophage or plasmid infection. They were later discovered and developed as a programmable gene editing tool. For gene editing, a guide RNA can be artificially synthesized. It will guide the Cas protein to the site it needs to be edited. The RNA in the Cas protein then can be delivered directly into the cells by microinjection in small organisms or transfection into larger organisms like humans. Once inside the cell, the RNA protein complex searches for the DNA sequence that is complementary to the guide RNA. Once the sequence is found, Cas9 protein then binds to a nearby PAM site on the DNA, which initiates the cut. The protein unwinds the DNA, and two domains of the protein each cut one strand of the DNA, creating a double-strand break. The cell will then try to repair a double-strand break through two methods. Either the broken strands are simply joined together, resulting in deletion or insertion, thus silencing the gene, or the cell will repair the break using another DNA strand as a template. A template containing new sequences can be synthesized and introduced into the cell. Now the cell will make the repair accordingly, and the new genes are integrated into the genome.
Scientists could use this technology to make changes in human genes. CRISPR Cas9 could be used to repair disease-causing mutations using a healthy wild-type copy of the gene as a repair template. To make disease resistance heritable, for it to be passed down between generations, scientists, and maybe eventually clinics, could alter genes in embryos, or the egg and sperm that formed them, meaning every cell in the person that embryo becomes will have the edited, resistant copy of the gene. A group of researchers in 2017 successfully completed genome editing in embryos, correcting a gene responsible for the inherited heart condition HCM, or hypertrophic cardiomyopathy. They took sperm from a patient with the mutation in a dominant HCM-causing gene and injected it into a wild-type egg, with CRISPR Cas9 to repair the mutation. They produced corrected wild-type embryos for the gene, a successful repair. However, these embryos were never implanted, so their long-term viability may still be in question. While this works in theory for simple one-gene diseases, things become much more complicated when we start to consider diseases or physical characteristics which are influenced by many genes, such as diabetes, Alzheimer's, or eye color.
What are the ethical and social implications in favour and against heritable genome editing? Heritable genome editing gives us the potential to cure very harmful and damaging genetic diseases. This could reduce the rates of certain diseases and their burden on the broader community and the healthcare system, as well as individuals. We would also be giving couples the opportunity who otherwise would not be able to have genetically related and healthy children. The question that arises from all of this is, do we have a moral obligation to cure all the disease we possibly can? We must consider the harms this technology could bring. There are potential safety and efficacy issues which bring risk and unknown long-term consequences for the individual. There are also adverse psychological effects that being edited could have on our children, as well as social stigma that could come from the technology. There is the issue of justice. Who will get access to this technology, and will it further the divide between the rich and the poor? Should we be redesigning our fundamental biology as humans, and could this lead to genetic enhancement? The question is no longer can we, but should we?